Now students, in today's lecture, we're going to discuss the differences between sickle cell anemia and thalassemia. So, these two diseases that are sickle cell anemia and thalassemia are basically categorized under a heading that is hemolytic anemia. So, what actually happens in these types of anemias? Basically, in it, there is destruction of RBCs. We have unhealthy RBCs. We have RBCs with short lives. So, this intrinsic hemolytic anemia is inherited disease. So, two main examples of this intrinsic hemolytic anemia are sickle cell anemia and the thalassemia. So, what happens in both these examples, what happens in both these diseases, we have abnormal shape of RBCs. Secondly, we have fragile RBCs, which are susceptible to hemolysis that can easily break down. So, what are the basic difference between these two diseases? Firstly, discussing the sickle cell anemia. Like thalassemia, sickle cell anemia is also an inherited disease in which we have RBCs which are in sickle shape, that are in moon shape, that are crescent shape. From the center, the cell is thicker, while from the edges, we have thin edges, we have tapered edges. So, in sickle cell anemia, we have RBCs which are moon shaped, which are crescent shaped. So, this is basically an inherited genetic disease. What happens in this case, the individual will inherit a defective gene, will inherit a gene that is hemoglobin S from both of the parents. He will inherit copies of hemoglobin S gene from both parents, that is from mother, from father as well. Okay. Now, the infected person has hemoglobin SS. So, it means he is suffering from sickle cell anemia. Other name of sickle cell anemia disease is hemoglobin SS disease because the particular individual has hemoglobin S gene from mother and hemoglobin S gene from the father. Now, this is inherited disease. Now, what this hemoglobin S causes? This hemoglobin S gene actually results in defect in the beta globin chain of the hemoglobin. We know that hemoglobin contains two alpha globin chains and two beta globin chains. While in case of sickle cell anemia, what happens because of defected gene, because of hemoglobin S gene, there will be a defect in the amino acid sequence. There will be substitution of valine for the glutamic acid. So, this will particularly happen at position number 6 of the beta globin chain. So, this was a basic and the scientific reason behind the sickle cell anemia. So, in this case, we have abnormal hemoglobin, that is hemoglobin S, because we have a gene that is hemoglobin S gene from both of our parents. So, in this, the alpha chains are quite normal, but the beta chains are abnormal because of the change in sequence of amino acid at position number 6. So, the molecule of hemoglobin S will polymerize into the long chains, and these long chains will precipitate inside the cell. So because of this, the RBC will attain a sickle shape now because we have precipitated chain in the cell. So the center is the crescent shape, is a thicker shape, and the edges are the thin shape. These are the tapered ones. Now students, in sickle cell anemia, basically we have cells which are in the crescent shape, that is thin edges, but the middle is the thicker portion. Now, in sickle cell anemia, we have noticed that we have two abnormal genes, one from each parent. So, we have RBCs that can get easily hemolyzed, that can undergo easy breakdown. And now these sickle cells have ability to block the blood vessels. So now these blocked blood vessels can lead towards the infarction, and we know infarction is stoppage of the blood supply. So that infarction will particularly be common in small bones, particularly of hand and the foot. And ultimately, there is possibility of varying lengths of the bones of the hand and the foot. There will be varying length in the digits. So, this condition is called the hand and foot syndrome. As we know, there is destruction of RBCs. So, we know whenever there is destruction of RBCs, there is release of bilirubin. So this bilirubin can result in jaundice in these children, in these patients. Now, this was all about the sickle cell anemia. Now coming towards the thalassemia. Thalassemia is an inherited disorder like sickle cell anemia in which we have abnormal hemoglobin. It was previously known as Cooley's anemia because he was a scientist who presented the clinical features of heart disease regarding thalassemia. So, this is quite common in Mediterranean cities, countries like Spain. So, while the sickle cell anemia was common in Africans. So, this thalassemia is most common in Thailand, in Mediterranean countries. There are two types of thalassemia. First one is alpha thalassemia, second one is beta thalassemia. So major difference which you should remember between sickle cell anemia and thalassemia is that in sickle cell anemia, we have beta globin chains which are basically affected, while in case of thalassemia, we have both alpha and beta chains which are affected. Now, coming towards the detail, but before this, you must know that beta thalassemia is most common. In normal hemoglobin, we have normal alpha and beta chains, while in thalassemia, the production of these chains become imbalanced because of the defective synthesis of the globin genes. So what happened in the thalassemia? Same, there will be precipitations of the polypeptide chains in the RBCs. There will be not a proper shape of RBCs. We will have immature RBCs that will undergo hemolysis. So what happens? This will lead towards the disturbance of the RBC's production. So basically what is happening in thalassemia, there is precipitation. Precipitation may possibly occur in the mature cells. So ultimately what we are getting RBCs which can get easily lysed, which can easily undergo hemolysis. Coming towards alpha thalassemia. Alpha thalassemia occurs in the fetal life. First thing, this is common in infants. This is common in infancy. In this, the alpha chains are less, absent, or abnormal. This alpha chain feature is associated with alpha thalassemia. In alpha thalassemia, we had affected alpha chains. No doubt beta chains are also affected. There is possibility that in adults, the beta chains are in excess, and in children, we have the gamma chains in excess. So this leads towards the defective production of RBCs, defective erythropoiesis. This will lead towards hemolysis. The infants in this case may be stillborn, that is death or loss of baby before or during delivery, that is stillborn, okay? Or your infant may die immediately after the birth. So this was all about the alpha thalassemia. Basically, I have discussed about the fetus. In fetus, we have the fetal hemoglobin. This is the dominant form of hemoglobin which is particularly present in the fetus during gestation time, that is from the 9th week until birth. So this period from 9th month till the birth is the fetal period, and in fetus we have fetal hemoglobin. So in normal cases, in adults, we have normal hemoglobin, 2 alpha chains and the 2 beta chains. While what happens in alpha thalassemia, there is decrease in amount of alpha chains. We also have unstable beta chains as I have discussed in my previous slide. So this will all cause the increase in destruction of the RBCs. Now, what are gamma globin chains? The genes which codes for these are the HBG1 and the HBG2. So these are normally the genes which are expressed in the fetus. So two gamma globin chains together with the 2 alpha chains constitute the fetal hemoglobin, that is HbF. While this is replaced in adult type, which is normally replaced by the adult hemoglobin, that is HbA. So this was all about the alpha thalassemia. Now coming towards the beta thalassemia. What happens in beta thalassemia? In beta thalassemia, the beta chains are less in number, absent, or they are abnormal with excess of gamma chains. So both chains are affected in case of the thalassemia. The alpha chains precipitate, causing defective erythropoiesis and hemolysis. So this was all about the differences between the sickle cell anemia and beta thalassemia. In next lecture, we will discuss other types of the anemia.